There are 3 normalization strategies to evaluate for compatibility with the biology, context, and the goals of your experiment. The first and most well-known is the housekeeping protein strategy. A single common housekeeping protein, such as GAPDH, actin, or tubulin, is chosen as the internal loading control. For this internal loading control to be accurate, it must adhere to the characteristics we talked about earlier. That is, it must be an endogenous protein present in all samples. The proteins expressed by housekeeping genes are generally involved in cell maintenance and basic functions and are constantly required by the cell. Secondly, it must be unaffected by the experimental conditions or treatments. Although expression of housekeeping proteins can be stable, recent studies have shown that experimental conditions can alter their expression. The Journal of Biological Chemistry's submission guidelines state, housekeeping proteins should not be used for normalization without evidence that experimental manipulations do not affect their expression. This means you must provide data from a validation experiment that shows expression of your chosen housekeeping protein with and without the change in the experimental conditions. These conditions are not limited to drug treatment, but may also include biological issues such as tissue variation, environmental factors, and developmental timing. For example, here a lab was looking for a stable loading control and found that actin and tubulin were not stably expressed during embryonic to postnatal retinal development, but MAPK was. Finally, can the housekeeping protein be detected in the same linear range as your target protein? Housekeeping genes are well known for high protein expression levels that can result in saturation of bands, particularly when chemiluminescent detection is used. Signal saturation is a significant and frequent problem in normalization that occurs when the signal intensity from a band is too bright for the detector to accurately record. When this occurs, signal intensity is no longer proportional to abundance. Saturation causes strong bands to be underestimated, concealing variation between the bands and causing loss of data that will affect your data analysis. Here is an example of how rapidly saturation occurs on film. Only 3 bands are in the linear range of detection. The last 4 are not. The signal should be shooting straight up, but instead you see an abrupt plateau, indicating that the detector, meaning the film, cannot differentiate the variation in the signal from the increasing amounts of the loaded target. This means the data are lost. The 4 saturated bands you just saw have been graphed and seem to have similar optical density values due to the saturation level or ceiling. However, if the same data are captured using a different type of detector where the bands can be detected, the recoverable data would make a radical difference in your analysis. Often, to detect the target protein at endogenous levels, large amounts of cell lysate are loaded, resulting in extreme overloading of the abundant internal loading control, as I just mentioned earlier. When the abundance of the target and internal control are quite different, as shown in this graph, the linear range of detection and optimal sample loading will also be very different. This discrepancy makes quantitative analysis much more difficult and inaccurate. For any normalization strategy you choose, you must verify that your internal loading control and target protein concentration are within their combined linear range using your specific antibodies, sample concentrations, and detection method. With housekeeping proteins, the type or origin of the internal loading control protein chosen should also match the target. For example, if the target is a mitochondrial protein, then the housekeeping protein should be as well.